can go ahead and get started. So welcome to the first iOS STN seminar of the fall. We're very excited to have Robin Tinghitella and Phillipp here today to talk about sensory ecology and speciation. Before we get started, just a quick note about our next seminar. So the next seminar topic will focus on neurobiology and behavior in speciation. It's going to be held the 15th of October at 11 a.m. Central European time. So please mark your calendars for that seminar, and there will be updates on our mailing list as well. So we can go ahead and get started with our first talk, which is from Robin Tinghitella. So it's my great pleasure to have Robin here today. Robin is an associate professor at the University of Denver. Her work spans a broad array of topics, including the role of behavior in rapid evolutionary change, the contributions of sexual selection to speciation, and the interplay between different selective pressures shaping the evolution and diversification of animal signals. Robin asked these questions in a variety of study systems, including the three-spine stickleback and the Hawaiian field cricket. To me, Robin's work has always really been characterized by very insightful questions coupled with just really elegant and integrative experimental designs. And so in particular, her recent work on the origin and spread of a novel sexual signal in Hawaiian crickets is just an extraordinary example of how we can study the contribution of sexual signals to population divergence and the formation of reproductive barriers. So we're really excited to have Robin here today talking about the origins of novelty in animal communication. So Robin, whenever you're ready, please share your screen.
Okay. All right, how's that looking? Awesome. Well, thank you so much for the invitation to talk with you all today and for the lovely introduction. Our topic for today is sensory ecology and speciation. So what I want to do is start with a bit of background and then relate that background to the work that we do in my lab to understand the origins of really truly novel sexual signals. So some of the most spectacular and diverse traits across animals are the signals that are used to attract mates. And closely related species often differ dramatically in signaling traits, despite widespread similarity in other characteristics. I'm showing you some examples of signals used in mate attraction here that differ between closely related species across a variety of taxa, from treehopper vibrations to cichlid colors to cricket songs. And I want to dig into that cricket song idea a bit in the talk today. Of course, the idea that divergent mating signals and corresponding preferences can create a really potent barrier to gene flow during early stages of speciation is an old one. And recent comparative and experimental studies offer support for this longstanding idea. I think crickets have really proven to be a powerful system for asking questions about that process. Male crickets produce songs that function both in species identification and in mate choice, and they do that using the wing instrument that's pictured here. So they rub the file of one wing against the scraper of the opposite wing, resonating structures like the harp and the mirror to produce those characteristic chirps that we're all familiar with. For instance, native Hawaiian crickets in the genus Laupala produce mating songs that have species-typical pulse rates. This figure shows the pulse rates of two Laupala species that are described as ecologically indistinguishable. They're cryptic species, except that their calling songs have characteristically different pulse rates. One is slow in the black bars, and one is fast in the gray bars. In parallel, then, females have preferences for conspecific pulse rates. The fact that the song and the preference are conspicuously different between species, while other traits aren't, suggests that sexual selection is a primary mechanism contributing to rapid speciation in this group. So what does that all have to do with sensory ecology? Well, ultimately, understanding the causes of these differences in signaling traits and preferences that form pre-mating barriers to reproduction would really illuminate how sexual selection operates within species and how reproductive isolation evolves. Sexual signals are often complex stimuli that integrate multiple sensory components, and each of those is produced by underlying morphology, physiology, or behavior of that signaler. So I'm going to conceptualize that today as the instrument pictured here. That instrument is going to generate a signal that's broadcast out into the environment. And of course, signal characteristics could be arbitrary, but they could also be shaped by selection. Assuming selection is important, what's likely to drive the evolution of these signals? Well, to be effective, signals need to propagate successfully through the environment, and the receiver needs to be able to detect and distinguish them from other sensory stimuli. So how signals and receiver perception interact with the environment is really important to this process. I'm also going to emphasize today that natural enemies like predators and parasitoids that eavesdrop on sexual signals might have an important role to play in diversification for two reasons. Eavesdropping enemies could select directly on signals, and they could act indirectly through their influences on the sensory systems and signal processing of intended receivers. In the early 1990s, two hypotheses were proposed that posited sort of mechanistic underpinnings for how animal communication might be influenced by sensory systems to evolve in one direction or another. When a signal is produced, a receiver first has to be able to detect it using sensory organs on the background of whatever the environment is. And anything that's outside the receiver's range of sensitivity is essentially undetectable. It's invisible, inaudible, and odorless. Often, greater sensory stimulation translates into stronger preference, but how that plays out when the receiver is making a decision depends on perceptual processing, and that isn't always straightforward. There have been a number of recent calls for better integration of sensory and cognitive mechanisms of receivers into our understanding of the evolution of animal communication. Now, the sensory exploitation hypothesis, one of those two, posed that sensory systems have properties that affect how stimulated they are when detecting and transducing a particular stimulus. For the most part, receivers prefer signals that are perceived as brighter, louder, and smellier, and signals can evolve simply because they better stimulate that receiver. Sensory drive built upon that idea. This hypothesis predicts that both male mating traits and the perceptual systems that underlie female preferences adapt to local environments. So divergence occurs because local environments impose selection on signals and sensory systems, and the result can be qualitatively different mating signals and preferences across environments, which of course could lead to sexual isolation. How would that work? I'm going to use three-spine stickleback as an example here, and this is largely work by Jenny Bauman from the early 2000s. The freshwater lakes that stickleback live in can differ in their transmission properties. In some lakes, breeding grounds have that typical bluish-greenish water color, but in others, the water is red-shifted in color. And as you might imagine, the stickleback's bright red throat should be masked in those habitats because of reduced contrast of that signal against the background. Accordingly, the throat color of male stickleback that live in red-shifted light environments has actually shifted from red to black coloration. In other words, signals came to vary with their habitats. Males now have colors that are likely to contrast with the habitat and thus transmit to receivers readily. The next step here would be for perceptual sensitivity of choosers to also vary with habitat, and for the signals in each habitat to match that sensitivity. And indeed, female sensitivity to red light in the eye is reduced in populations that nest in these dimmer, red-shifted water habitats. And male signals match that spectral tuning. The black coloration stands out well in a red-shifted habitat, given the female's visual sensitivity. Next, then, we expect for female preferences to vary with their perceptual sensitivity. And Jenny found that across replicate population, the extent of preference for red depended in part on perceptual sensitivity to red. In other words, females preferred the reddest males when they have lower detection thresholds for red. And then finally, a critical prediction is that mate signals and preferences should diverge, and that divergence should contribute to reproductive isolation. Here, Jenny was able to show that populations of stickleback that differed most in their signals, that's on the x-axis, were less likely to spawn with one another on that top graph. And in the bottom graph, populations containing females with more divergent preferences were less likely to spawn with one another. Okay, so logically that all makes plenty sense, but I still have some questions about how populations come to differ in male signaling traits to start off with. Because at any one point in time, we actually expect receivers to be attuned to and to have strong preferences for the signals that already exist locally. So when a novel trait initially invades, and it's very rare, virtually all individuals of the opposite sex should discriminate against them, making it difficult for new sounds, smells, colors, and dance moves to get a foothold. So today, I want to tell you about a study system in which we're getting to watch that process directly in real time, and I think we're gaining some new insight. For the last 20 years or so, I've been studying the evolution of sexual signaling in the Pacific field cricket, Teleogryllus oceanicus. These aren't the native Hawaiian crickets that I told you about earlier. Instead, this is a really broadly distributed cricket. It's native to northern and eastern Australia, and it's island hopped through the Pacific. Most recently, the cricket was introduced to the Hawaiian Islands when they accompanied the first human arrivals on outrigger canoes. So in our system then, the cricket's wing is the instrument, and it produces a signal, a loud, long-distance song. That song sounds like this. And it's subject to lots of different selective pressures, including intended receivers, our female crickets, transmission properties of the environment that influence which signals successfully reach those receivers. And in Hawaii, but only in Hawaii, a deadly eavesdropping parasitoid fly called Ormia ocratia. That fly eavesdrops on the songs that male crickets produce and uses them to locate hosts. They sort of spray their larvae on singing crickets, and then the larvae will burrow in and feed on the cricket's insides for about 7 days before they emerge, pupating and killing their cricket hosts in the process. So today I want to ask how and why that cricket's song, which is generated by that wing instrument pictured here, has managed to diversify over just the last 20 years, generating a rainbow of variation in signals in Hawaii. And then think about whether that song diversification is actually likely to lead to speciation. So this story begins sometime between 700 and 1600 years ago when the cricket was introduced to the Hawaiian Islands. We don't actually know how long they've co-occurred with the eavesdropping fly, but the first record of the fly in Hawaii is from 1989. Shortly after that, Marlene Zook, who was my PhD advisor, started studying the interactions between this cricket and fly and had continuous field studies running throughout the 1990s. During that time, all of the crickets had this typical wing type and produced what we now call the ancestral or typical song that sounds just like the one I played for you a couple of slides ago. That song has a really characteristic temporal pattern, and it's loud and relatively tonal, so it travels really well through the grassy fields where these guys are singing at night. Now, if you know anything about this study system, what you probably know is that in 2003, we discovered silent male crickets on the island of Kauai in Hawaii. These males went silent because an X-linked wing mutation wiped out many of the structures on the wings that are used to produce song, leaving the wings flat. So you sometimes hear them called flat-wing males. Not only that, but they spread through the population from negligible to more than 95% of males in fewer than 20 cricket generations. And that's because being silent actually protects these males from that parasitic fly. And then things were quiet for quite some time until 2017, when I traveled to the island of Molokai. And there I found that the crickets were actually singing a brand new song. I called it purring because I initially thought that the strange sound I was hearing must be coming from some nearby stray cat. The wings of purring males are really similar to those of silent males. They're missing a number of important structures and they have really reduced resonators. But purring males are capable of producing sound, and the songs that they sing bear little resemblance to those ancestral typical songs. Purring males are quieter, the song has much higher frequency, and it's much more broadband than that ancestral song, which gives purring males a sort of noisy quality. So here's what that sounds like. There's one major way that purring male wings actually appear to differ from silent male wings, and that's that the majority of purring males have a scraper and silent males don't. The scraper is this really little organ on the cricket's wing that is rubbed against the file to resonate the wings and produce that sound. And we recently found that whether you have or don't have just that tiny little structure on the wing is enough to change the detectability of songs, both to female crickets and to us. So this figure is a stylized cricket audiogram showing how loud a sound has to be to elicit a response from a female and how that depends on the sound's dominant frequency. So everything that's shaded in gray here is a region of acoustic space that would elicit a response from a female cricket. Now, the presence of a scraper on a purring male's wing actually shifted the peak frequency of song from about 14 kilohertz, which is not very detectable to a female, down to about 8 kilohertz, much more detectable to female crickets and also to researchers. In fact, tones with peak frequencies that match those of males who don't have a scraper would need to be about 19 decibels louder to elicit a response from a female than those that are produced by males with scrapers. So armed with that information, we continued monitoring cricket and fly populations over the next few years, visiting Hawaii every 2 cricket generations or 6 months for really standardized studies. And just 2 years later, in 2019, a graduate student and a postdoc in the lab, Jay Gallagher and David Zanana, started to hear things in our data that they couldn't explain. Songs that didn't neatly fit into those already described categories of purring, silent, and typical. And that's because there was yet another new morph. This one we called rattling, and it's only found on the Big Island of Hawaii. Here's what rattling sounds like. Like purring, those rattling songs are higher in frequency, more broadband, and quieter than the ancestral song. But unlike purring, the wings of rattling males are nearly identical to those of typical males until you really examine the file under a microscope. We found that the file of rattling crickets has characteristic gaps in it where teeth are missing, and that change dramatically changes signal properties. Meanwhile, also in 2019, Nathan Bailey's lab published a paper reporting 2 more novel cricket morphs in Hawaii, each of which produces yet another different signal. One of those, called small wing, has miniaturized wings, and it was discovered on the Big Island. And the other, curly wing, has wings that sort of curl up on the edges, and that one was discovered on the island of Oahu. So having found several male types that each generate these different signals that all seem to be evolving over a really short time span, we wanted to dig in and ask how it was that each morph was balancing the conflicting natural and sexual selection pressures in Hawaii. So we addressed these questions using field-based phonotaxis experiments to capture natural mate and host location behaviors by broadcasting different types of song and white noise negative controls from speakers that we placed out in the field. So on this figure, I'm actually summarizing a huge amount of data that we reported across about 4 different papers. On the top is the percentage of females attracted to each signal. And on the bottom is the percentage of parasitoid flies attracted to each signal. So on the far right here is the typical song. It's very attractive to females, but also very attractive to parasitoids. And on the far left is the silent or flat-wing type, which escapes parasitism, but also has a really difficult time attracting mates. The novel signals are in the center of this figure. And largely, they all seem to be at least moderately attractive to females, despite some of them being relatively quiet and broadband. And each of them protects their bearers to a large degree from detection by that parasitoid. So some of these novel signals then seem to allow private communication among crickets, at least for now. More recently, things continue to change out there. We just characterized yet another type of male that we're calling broken harp that we started noticing in 2019. And we'll keep watching and hopefully be able to fill in some more of the story along the green arrow here in coming years. So naturally then, we were really curious to know what exactly was facilitating all of this. Why are these novel songs successful despite having such different sound characteristics? How will they evolve moving forward? And will preferences arise and become coupled with those signals? The list of questions just goes on and on. Much of that is going to depend on receiver psychology. Most animals have a pretty broad sensory capacity that allows them to detect and respond to cues of conspecifics, predators, and prey, for instance, although they may only respond positively to trait values that match their preferences. T. oceanicus, the cricket, is capable of hearing sounds that range from about 2 to 100 kilohertz. So on the left, I'm showing you a behavioral audiogram that shows the amplitude at which crickets respond to tones that vary in frequency. So the lower that black line is, the more sensitive the animal is at that frequency. Crickets are most responsive at 5 kilohertz, which makes sense because that's the dominant frequency of male songs. But notice that they can actually hear quite a lot outside of that range. Similarly, on the right is a fly neural audiogram in light pink, indicating the amplitudes at which flies from Florida, where they're native, can detect each frequency on the x-axis. Auditory tuning of Floridian flies also peaks at 5 kilohertz, matching the song of their local host cricket. But again, those flies can hear quite a lot more than just the dominant frequency of their host songs. So when we initially found purring crickets, we decided that we wanted to do a really deep dive into the nitty-gritty of crickets' preferences. In essence, we wanted to know, is beauty in the ear of the beholder? So we first looked at preferences of crickets and flies for song frequency. This figure is showing the responses of female crickets on the y-axis to manipulated purring songs on the x-axis that differ just in their frequency or pitch. And those thick dashed lines that you're looking at are preference functions that we aggregated at the population level. So for crickets, there's this really clear prominent pattern. Those aggregate preference functions are nearly flat, regardless of which metrics of female response we measured. In other words, we found no evidence that your average female cricket prefers certain purring songs over others. And intriguingly, although flies don't seem to locate purring songs from afar, if you bring them into close proximity with purrs, some of them can. And that pattern I just described of flat preference functions is also true for flies. But those flat preference functions are driven by some pretty incredible interindividual variation in responses of individual crickets and flies. So the light lines here in the background are the preference functions of individuals, and they are all over the place. Some respond to all purrs, some respond to none, some have really extreme preferences, and some have intermediate preferences. And these results are incredibly robust, regardless of what component of song variation we put or vary on that x-axis, we get similarly flat preference functions. So that might seem strange, but this was actually a pattern that I had seen before. When I was in graduate school and we discovered silent crickets, I did this big courtship study where I was simulating secondary contact between typical crickets from across their range and silent males from Hawaii. And what I found was that females from island populations in the black bars, and specifically from Hawaii on the far right of this figure, were much more willing to accept silent males as mates. They seemed to be really permissive in the context of mating, which allowed silent males to achieve some reproductive success. We've now found that pattern of relatively relaxed mating preferences in Hawaii in other contexts, which leads us to hypothesize that permissive preferences might actually be an important hallmark facilitating the earliest stages of novel signal evolution. After all, if female crickets were incredibly choosy about their mate's songs, new songs like purring and rattling would never become common within populations. But on the flip side, that permissiveness might make it less likely that you get coupled signals and preferences that differ across locations and contribute to reproductive isolation. Unless there are some changes in receiver psychology. Everything I've just shown you sort of implicitly assumes that sensory systems aren't evolving, but what if they did? We haven't looked at this in Hawaiian crickets yet, unfortunately, but we have in flies. And this is work that was done in collaboration with Norman Lee at St. Olaf College, who is a neurophysiology whiz and who really made all of this possible. We used complementary experiments that probed the auditory tuning and the behavioral thresholds of flies from two locations, one ancestral population in Florida where the flies are native, and one Hawaiian population where the fly was introduced. So to test for evolved differences in the overall auditory sensitivity, we recorded extracellular neural activity from the neck connective of flies from those two populations in Common Garden. That neck connective contains auditory neurons that relay input to the brain. And we tested responses to pure tones that varied in frequency from about 2 to 40 kilohertz, that's on the x-axis, at sound intensities from 20 to 90 dBSPL on the y-axis. And the super cool result here, I think, is that the shape of the fly's neural frequency tuning curves differed across populations, with Hawaiian flies being more sensitive to frequencies around 5 and 10 kilohertz. Those frequencies correspond to the dominant frequency of typical and purring T. oceanicus songs. Similarly, when we tested for evolved differences in the behavioral response thresholds of Hawaiian and Floridian flies using phonotaxis on this cool spherical treadmill system, we found that Hawaiian flies were more responsive overall, and particularly to frequencies that aligned with the typical and rattling T. oceanicus songs. So finally, we played some naturally recorded purring, rattling, and typical songs to flies from both of these populations, and again found that Hawaiian flies were overall more responsive to all three of those T. oceanicus songs. So I think taken together, these results really suggest that the neural auditory tuning and the behavior of Ormia have evolved in Hawaii in ways that likely facilitate the detection of these novel host songs. Okay, in summary then, we've seen that there's been evolution of at least 6 novel male morphs over the last 20 years. Each protects crickets from an introduced natural enemy. Song evolution might be facilitated by these sort of relaxed mating requirements in Hawaii. We don't have any evidence yet for preferences for specific novel signals. But natural enemies might actually be responding in this sort of game here through evolution of both auditory and behavioral thresholds that facilitate them locating males who sing novel songs. With that, I want to quickly say thank you to all of these people and funding sources, and I would be happy to take any questions you might have.
All right. Thanks so much, Robin. That was fantastic. We can take questions out of the chat, or if anyone wants to raise their hand, please feel free to jump in. Well, I can start then. So Robin, it seems like this variation in permissiveness is really important for populations to be able to shift between different signals. So do you have but it also, the trade-off is then that you don't get that coupling, or you don't get sort of specialization on specific signals. So are there scenarios where you think permissiveness would be selected for versus detrimental?
Yeah, that's a great question. You're reminding me of 2 things. So one is that I think this permissiveness might actually have evolved in Hawaii in particular because of something associated with the process of island colonization. So if you imagine that you are one of the few males or few females who arrives on a new, island, not very many individuals made it there, and you are trying to find a mate, if you're extremely choosy about the mates that you are willing to hang out with, you're actually probably not going to find anybody who's acceptable, right? So there's this idea from Ken Kaneshiro that process of island colonization actually selects for females who are less choosy in terms of, what they require for mating. So I think there are lots of situations like that. That's a very specific one associated with island colonization, but you could imagine having issues with locating appropriate mates in a lot of different scenarios when you've got fragmented habitats, small populations for whatever reason. There's also all this cool work that Nathan Bailey's done looking at how the acoustic environment, what the crickets actually hear, influences how choosy they are. So you can actually even further relax those mating preferences depending on what you've been experiencing in the environment. So I think there are a lot of reasons that you might end up with a situation where you've got permissive preferences, but really, so is that going to mean that you don't end up getting this sort of coupled preference and signal eventually? I don't know, right? Because one of the hallmarks that seems to underlie those really permissive preferences at the population level is this extreme interindividual variation. So if we were able to find situations where females who do have a preference for a particular type, if there are a few females who really like this type of purring in particular, you might actually end up getting some coupling. It only really takes a handful of, individuals who have that particular type of signal and individuals who like that particular type of signal. So theory suggests that this could work, particularly because there's all of that inter-individual variation. And we're really interested to see eventually when we do some cool neurophys with Norman with our crickets, we're interested to see if what underlies that variation in individual level preferences is something about individual variation in sensory systems, right? So there could be good reasons that we're seeing all of that variation.
Awesome. Thank you so much. We'll move on to Philip now, but we can come back to any other questions for Robin when we move into the general discussion. So thanks so much.
Yeah.
All right. So it's my pleasure to introduce Philip Brand, who is currently a postdoctoral researcher at Rockefeller University. So I first got familiar with Philip's work at a GRC conference a few years ago, and it just really impressed me because of how integrative it is. So he uses methods from molecular evolution, functional genomics, and behavioral studies to understand how diverse insects signal and respond to each other, and how that can then lead to species divergence. And what's also really impressed me about Philip's work is that he's extending methods developed for model organisms to a broader range of critters, like my favorite, the orchid bee. And I can imagine that's not easy. So technically, the work he's doing is just really impressive as well. And in so doing, what I also really respect about this work, it's maintained a really deep grounding in the natural history and ecology of the systems he studies. So Philip, thank you so much for joining us, and I look forward to your talk.
Yeah, thank you so much for this very kind introduction. Let me share the slides. Can you see this? Okay. So I would really like to thank Liz and Joanna for the invitation, and also the entire iOS committee for putting this together. It's very exciting to be here. And in this video that I'm showing you here, you can see that the behavior of each individual fly is very much dependent on its immediate environment, making them switch between feeding and interacting with other flies quite rapidly in different ways. And now, yeah, this works. This ability to respond appropriately to the environment allows animals to coordinate their fundamental needs to survive and reproduce. And I'm interested in this extraordinary adaptability that is enabled by the nervous system, but not over the lifetime of an animal, but over longer evolutionary timescales, because evolution has provided this incredibly vast diversity of behaviors across the animal tree of life. And for example, each of these species that I've put on the slides here have evolved very different ways to exploit the diverse food sources that they rely on, and also to find mating partners engaging in the social interactions that are necessary to successfully reproduce. And this behavioral diversity of the nervous system is encoded in the genome. So what fascinates me, and the overarching question that is driving my work, is how evolution shapes genomes and nervous systems to generate behavioral variation. And then in the next step, how does behavioral evolution impact the evolution and maintenance of reproductive isolation? So today I will be talking about two aspects I've been working on. First, I will briefly touch upon work that is all published that I did on the genetic basis of perfume evolution in orchid bees, and then I'll transition and spend most of the time on what I'm currently doing on the neural basis of courtship evolution in fruit flies. Orchid bee males collect fragrances from flowers in their tropical environment in order to produce a complex chemical blend that we call the perfume. And in order to do so, the males require the sense of smell to find these fragrances. The perfumes they concoct are species specific, and they're used to attract females, which also rely on the sense of smell in order to detect the perfumes and choose males to mate with. So in order to understand how perfume communication evolves on a molecular genetic level, I began by characterizing the genetic underpinnings of the sense of smell and its evolution. And here I focused on the odorant receptors, which are integral to the sense of smell because they bind odors and enabling insects to smell. And it is a rapidly evolving, very large gene family with over 100 genes in any bee species, and in flies, it's about 60 to 70 receptor genes. Unfortunately, I don't have time to talk about this today, but if you're interested in this, please ask me later or contact me after the seminar. So after doing the groundwork on gene family evolution, I was now able to study perfume evolution on a genome-wide level across two closely related species of orchid bees. For this, I first went to the field and collected hundreds of bees throughout Central America, which I then used to conduct a population-based analysis of the chemical divergence of perfumes, the genetic divergence across species, during which I discovered odorant receptors that evolved extremely rapidly compared to the genetic background. And using electrophysiological techniques, I then was able to tie the molecular evolution of the receptor to its functional divergence, which allowed me to link the species-specific collection of perfume compounds to functional receptor divergence. And this was an incredibly fast explanation of what we did. If you're more interested, I welcome you to read a bit deeper into it or ask questions afterwards. Overall, what this work suggests is that receptor evolution is likely an important driver of perfume communication and differentiation and the evolution of new orchid bee species. So what was really cool about this project was it allowed me to go very deep on the receptors and the functional diversification, but it did not allow to understand how the preferences that males of different species innately have for compounds to collect are encoded. Because for this, I would have needed to go deeper into the nervous system. And although I tried, it's extremely hard in orchid bees. And so to be able to study behavioral evolution on a circuit level, I shifted for my postdoc at the work that I'm doing now to species that are more close to Drosophila melanogaster, a model organism, to look at the neural basis of courtship evolution. And what I hadn't appreciated before I started working on Drosophila is its fascinating diversity. I'm highlighting this by the variety of food sources that these flies occupy. Different species occupy different food sources, and on these food sources, they congregate in large numbers to feed and also to find and choose mating partners. And I became very interested in how these complex environments control courtship across species with different feeding ecologies and sensory ecologies. So what does courtship look like? In behavioral assays, like here with Drosophila melanogaster, males immediately engage in courtship when they encounter a female. And during courtship, the male persistently pursues the female for minutes. So the male is the individual following the other one, and he's just very hard trying to convince her to mate with him. And while these innate behaviors are highly stereotyped, they evolve very rapidly. All the species I'm showing you here perform their courtship in different ways, and they're closely related to one another. And to me, a species that stands out in particular is Drosophila erecta, which is the one I highlighted in green on the right side. It is kind of weird because it does not engage in courtship or mating behaviors in this assay. Instead, the male and the female are entirely uninterested in one another. And this is actually a video that I'm playing here, and I'm actually playing it 4 times as fast as the others. They're not interested. What I found, though, is that only when I add food into the acid chamber, erecta engages in vigorous courtship that is comparable to what the other species are doing without the requirement of food. And why that might be is maybe answered by differences in the host ecology of the species. While Drosophila melanogaster and actually all the other species that I'm showing here on this tree are broad generalists and able to exploit large varieties of food sources, Drosophila erecta feeds primarily on a single host, the fruit of the pandanus screw pines. And unfortunately, we know very little about this interaction from the field, but what we do know is that erecta evolved olfactory adaptations towards their host fruit, which includes an increased number of olfactory sensilla on the antenna and enlarged brain regions that are involved in processing the host odors and elicit species-specific behaviors when the host is present. So when I place a group of Drosophila erecta in a large arena with a small central food patch, what I find is that erecta rapidly congregates on the food on the left side, where they engage in vigorous mating behaviors, which contrasts with the agar control on the right, where flies are mostly apathetic of one another and don't engage in courtship. This is in stark contrast to Drosophila melanogaster, which courts vigorously, but does so throughout the entirety of the chamber. And this the way that erecta is attracted to the food patch becomes even more apparent from looking at the trajectories of animals over time that I'm projecting here on this slide. So this seemed to me a very intriguing example to study how the immediate environment may influence behavior and its evolution across species. We know that Drosophila melanogaster males engage in highly motivated courtship pursuit when they're sexually aroused, and they will even pursue unsuitable visual targets, such as here, these two-dimensional shapes that we're projecting onto the assay chamber. The males vigorously pursue and court, although they will never be able to mate with them. And it found that Drosophila erecta males will persistently court a visual target when food is present, which you can see on the left side. The male is very interested in this visual stimulus and is tracking it, whereas the males ignore the identical visual stimulus without food on the right side. And this suggests that Drosophila erecta males are sexually aroused by food, which is a requirement that I do not observe in Drosophila melanogaster. They're always court very vigorously. And I think this reflects the fact that olfactory pathways now play a critical role in controlling a male's arousal, because anosmic males, so males that can't smell anymore, which are created by knocking out essential olfactory co-receptors, these males do not engage in tracking behavior anymore, even when food is present. And I can use a few neurogenetic tricks to manipulate the activity of neurons in the brain of the flies. And when I optogenetically activate the olfactory sensory neurons in the antenna of erecta males on agar, I find that this is sufficient to drive them to vigorously court a female, which they usually would not do because of the absence of food. And I can even go a step further and present them with a plastic bead as a stationary visual target. And when I activate the olfactory sensory neurons, they become very interested in it and approach it over and over again, highlighting their visual arousal towards visual targets. So together, these experiments that I've been showing you suggest that in contrast to Drosophila melanogaster, erecta does not court just based on visual input. They actually require food odor, suggesting that food odor gates visual courtship pursuit in Drosophila erecta, but not in melanogaster and also not in other closely related species, the data I am not showing here. So how did this behavioral shift evolve on the neural level? We can study this very nicely in the fly brain because the neurons involved in male mating behaviors all express the same transcription factor that is called fruitless. And in the fly, we can label, access, and modify these fruitless expressing neurons, which I'm highlighting, which are highlighted here in green in the brainstem. And this makes this a very tractable system to study courtship on a circuit level. And recent advances in genome editing techniques now allow to translate the powerful neurogenetic toolkit of Drosophila melanogaster to its close relatives, which makes this a truly exciting time to study behavioral evolution because I am now able to examine it on a circuit level resolution between species. So I told you that Drosophila erecta depends on food odor to become sexually aroused. And in Drosophila melanogaster, sexual arousal is controlled by about 20 neurons that are called P1 neurons that are specific to the male brain, and they are a central node within the courtship circuitry in the center of the brain. And the role of the P1 neurons is conserved. When I activate them artificially in the fly brain, here in Drosophila erecta, this results in persistent courtship of visual targets, even in the absence of food, because I force the fly to be sexually aroused, which makes them track visual targets. And this visual behavior is reliant on a key population of neurons that is required for visual courtship. They're called LC10A neurons. And they are visual projection neurons that are small spot motion detectors that are tuned to the size and the speed of female flies. And LC10A neurons are essential for faithful courtship pursuit, and they're selectively active only when P1 neurons are also active, which ties LC10A activity and visual courtship pursuit to sexual arousal in males. So in order to understand how food odor may control sexual arousal, specifically in Drosophila erecta, this circuit node is actually a very intriguing starting point. To address this, I'm using a virtual reality system where I can examine courtship behaviors in tethered males that walk on a spherical treadmill, and I provide them with a very simple visual stimulus that oscillates from the left to the right horizontally in front of them on a conical screen. And then I also puff food odor with high temporal precision, and I then observe how the behavior and the neural activity changes in male. And this video here shows the male from behind. As you can see, he is not very interested in the visual stimulus in front of him. But as soon as food odor comes on now, he immediately starts tracking the target's movements very faithfully. And to me, this is a very satisfying video that I really love to watch. I could do this for hours. Well, but while the males perform this behavior, I can now image neural activity in the courtship circuitry by simultaneously imaging from the so-called lateral protocerebral complex, or the LPC, highlighted here in green, which is innervated by P1 neurons, and at the same time imaging from the AOTU, the anterior optical tubercle that is innervated by LC10A neurons. In Drosophila erecta, at baseline, the activity in the LPC that you can see above and the AOTU in the center remains unaltered. When the visual stimulus comes on, I see a slight increase in activity in both areas, but no tracking. The male is not interested in the visual stimulus, which you can see in the below graph. The tracking index is very low. When food odor is puffed now, neural activity rises and remains elevated throughout odor application, and males now persistently track the visual stimulus. So both neural activity and behavior after the odor application wane shortly after, which is in stark contrast to Drosophila melanogaster, where the visual stimulus is highly salient and males immediately engage in tracking, accompanied by high activity of the LPC and AOTU. So food odor seemingly has no effect on neural activity or behavior in this species. And the striking difference between the two species is highly robust across animals, which highlights the requirement of food odor for erector courtship, which is reflected in the odor-dependent activity of the courtship circuitry. So one thing that may happen here, though, is that we have a ceiling effect in Drosophila melanogaster, because visual stimuli are so salient that they may be already maximally aroused that we cannot see a clear effect of food odor anymore in the circuitry. So I repeated this experiment, but without showing a visual stimulus. And to my surprise, when exposing males to food odor without the visual target to court, both species show elevated activity during odor application. Which is interesting because it suggests that the ability to integrate environmental food odors in the central courtship circuitry is a conserved feature of both species' nervous systems, but it is required for courtship only in Drosophila erecta. And when I compare the relative activity of the LPC and the AOTU between species, I see no differences when males are courting during food odor exposure, which you can see here based on the heat map. But when there's no visual target, the erector AOTU is more active than the LPC, which is different in melanogaster, where the relationship is reversed, which you can see here. And this may suggest that there's a difference in how food odor is processed in the AOTU, meaning in the visual pathways of Drosophila erecta. So to look into this a bit more in depth, to understand where exactly food odor may be integrated in this circuit node, I now turned to the Drosophila melanogaster connectome to understand how P1 and LC10A are connected. So the connectome is the entirety of all neurons in the brain and how they're synaptically connected to one another. This is a great resource that has recently become available for Drosophila melanogaster, and now we're getting more and more other species, mostly of insects for full brains, to understand how these connectomes work and also diversify. It's a great resource. So Jerry Rubin's group at Genelia recently showed that LC10A receives, based on the connectome, receives strong inhibitory input. And these inhibitory neurons themselves are postsynaptic to inhibitory interneurons. This is a classic disinhibition mechanism that may release LC10A from inhibition. And indeed, they showed that P1 activity inhibits the inhibitory neuron that is presynaptic to LC10A, which means that P1 activity releases LC10A from tonic inhibitory input, which means that LC10A is now free to control visual courtship pursuit in aroused males. And to me, this provides an intriguing model to think more about where food odor may be integrated in the circuitry. And I imaged from P1 neurons and LC10A neurons directly in both species, but I didn't find any evidence for an effect of food odor, suggesting that it may be integrated in these disinhibition loop. So I now turn to Drosophila melanogaster, which I have shown is also able to integrate food odor. And I found that the so-called 2,2 neurons, which are presynaptic to LC10A, are being inhibited during male courtship pursuit and also by food odor, which you can see by this additional deflection of activity when the food odor in orange comes on. And this inhibition during courtship we would expect based on the disinhibition paradigm, but the response to food odor was new and very, very exciting, because when I imaged the neuron without a visual stimulus, it is very robustly inhibited throughout odor application. So this suggests that this neuron may be involved in integrating food odor into the courtship circuitry, releasing LC10A off tonic inhibition, which may enable faithful visual courtship pursuit, in addition to P1 activity. And I'm now in the process of translating the tools into Erecta to investigate its role in the odor gating of courtship. Okay, to wrap this up and get back to the initial question I raised, how does the environment shape courtship across species? I showed you that while Drosophila melanogaster males engage in robust courtship behavior when visual stimuli are present, Drosophila erecta does not. Instead, they require the presence of food odors in order to do so. However, the courtship circuitry of melanogaster has the ability to also integrate environmental food odors. And this suggests that the underlying circuits are conserved in their ability to integrate environmental food odor, but they're diversified to evolve this gate that I discovered in Drosophila erecta. So together, these results indicate that sensory inputs are weighed differently across different species, leading to the striking behavioral differences with respect to environmental control over courtship. And this may add another pre-mating barrier in Drosophila erecta. We know that gustatory pheromones in courtship song are very important and very in close contact of flies, but the use of a host that is not frequented by many other species and the requirement of food odor for courtship may add another barrier for reproduction in Drosophila erecta. Okay. What I am looking forward to in the future is actually combining these two different ways of looking at behavioral evolution from a genetic and a neural perspective to generate a mechanistic understanding of reproductive isolation. This, I think, would allow us to maybe tie peripheral diversification of sensory systems, which we in genetically focused studies, such as the one that we did on the orchid bee, um, may be more easy to understand if you're lucky enough and the genes that are involved are large effect loci that are also receptors that are which function is easy to understand. But this work that I'm doing in the flies and that also and other work from other groups suggests that there's a huge amount of variability in how multimodal sensory input is integrated with internal states to control reproductive behaviors. And this part is heavily biased by neuroscience techniques, and we do not have a good understanding of the genetic underpinnings. And I hope that in integrating the two, we can bring these more together and gain a better understanding of how behaviorals evolve and how they contribute to the evolution and maintenance of reproductive isolation. Yeah, with this, I would like to thank you for your attention. I would also like to thank Santiago Ramirez and the Ramirez Lab that work on the orchid bees during my PhD, and then Vanessa Ruta and the entire Ruta Lab for all the support that I have right now during my postdoc, also some funding sources. And if you find this interesting, please feel free to contact me whenever and chat. Yeah, thank you.
Right. Thank you, Philip. That was wonderful. I would welcome any questions via chat or if an attendee would like to raise their hand. Great. Sean.
Yeah, really fantastic stuff, incredibly detailed. I wish we could do this in other types of organisms as well. Just one of the questions I have. So the difference you see between Erecta and Melanogaster is really striking. How robust is that to, different selection lines or natural populations? How broadly have you tested for that effect within both species?
Yeah, you hit the nail right away. So first of all, I totally share your sentiment. One of the reasons why I switched to the flies is not so complex that it is actually it is possible to go to a circuit level, which are predetermined also genetically. So each individual has the similar circuits and neurons that are involved in behaviors. The second question is, unfortunately, there's only one Drosophila erecta line that is available that I've been using.
Incredible.
So I know it's not easy to do something like this when you come from a fieldwork PhD where you are able to sample throughout the entire distribution ranges of animals. The pandemic was also a reason why we couldn't do this. And so I'm now collaborating with a group in Cameroon where Erecta actually occurs to help and collect those flies. And it's very interesting because this association of Erecta with the Pandanus groupine is also a bit strange because when Erecta was described first, they found Erecta only in association with this fruit, but they only found fruit for 4 months of the year. And so nobody knew where these flies are.
Okay.
And our collaborators have done a first fieldwork to different sites where they talk to the locals. And at some sites, the locals say, yeah, the fruit is only available for a certain amount of the year. But in other sites, apparently they're fruiting throughout the entire year. And I would really love to go there and sample Erecta and Melanogaster and see what type of behavioral variation we can see with respect to this behavior.
Amazing. Thanks so much.
Thank you.
That actually relates to a question I had. I might have missed this, but was the food odor you were puffing in, is that Pandanus or is that something else?
It is actually something else because we also have the problem that we cannot sample Pandanus. What we use is apple cider vinegar. It is highly attractive to all fly species of the Drosophila melanogaster subgroup to which Erecta belongs, which to us was actually a good way to use a common food source for all these flies. I was lucky enough to get my hands on a little bit of Pandanus, and they respond in the same way to Erectodus responds in the same way to Pandanus. Where we think the difference lies is the long range attraction, where it seems like Drosophila erecta has adapted to be extremely good at finding Pandanus. So it could be that that this allows the flies to congregate on a common food source, and then they become aroused because they can smell food, and the food is actually not the fruit; it is the microbes that they find on the fruit that are decaying the fruit. So they eat yeast and other bacteria. And apple cider vinegar is a yeast fermentation product, and that's probably why they're so excited because it smells like food to them.
We'll take one last question from the chat, which is from Michael Ritchie. They're asking, what about the other specialist, Mauritania? Does it behave like Erecta?
So I think Mauritiana is not a specialist, but Drosophilus shellia is a specialist that is closely related. Maybe that is the species they're referring to. And for that species, uh, we know that it only occurs on the Seychelles and is specialized on Morinda citrifolia, which other flies cannot digest. And I have looked into one strain of Seychellia, and they did not show the same type of behavior. They also courted when there was no food present. So at the beginning, when I found this striking behavior in Erecta, I was very excited because it might have been such a cool way for specialists in general, but it seems like this is something that may be unique to Drosophila erecta.
Great. Well, thank you so much, Philip. That was a fantastic talk.